Enzymes are proteins that play a major role in the biochemical reactions happening every moment inside our bodies. Everything from digesting a bowl of ramen noodles to flexing your muscles in front of a mirror. Enzymes act as catalysts, meaning that they speed up the rate at which these biochemical reactions happen. So instead of waiting months to years for a reaction to happen, it can happen in seconds, which is essential for life to happen. Imagine trying to digest a single bowl of ramen for a year. You'd probably die of hunger before you could do it. Now, every biochemical reaction has a substrate and a product. So let's put them on this graph called a reaction coordinate diagram. The x-axis shows how a reaction progresses, while the y-axis shows the energy level at the different points along the reaction. In the beginning, we've got the substrate. Let's call it A, with a fair amount of free energy. At the end of it, there's the product, or B, which ranks lower, energy-wise. The energy of the product minus the energy of the substrate is called the energy of the reaction, also known as Gibbs free energy, or delta G. Because lower energy states are preferred, a reaction spontaneously happens when the product has a lower free energy than the substrate, so a negative delta G. So let's say we're looking at one such spontaneously occurring reaction. But between going from the substrate to the product, there's an intermediate transition step that has a really high energy state. The amount of extra energy the substrate needs to get to the transition state, so the height of the upslope, is called the activation energy, or delta G with a little double dagger symbol. As soon as it enters the transition state, and really wants to go to a more stable, lower energy molecule, so it either goes back to being a substrate or to being a product. If it's a substrate once again, it can go back up to the transition state if there's enough activation energy again. But if it becomes a product, then it needs even more energy to get back to the transition state. That's why over time, with millions of molecules doing this, the majority of substrate turns into product. Now, without an enzyme, the substrate might eventually harness enough activation energy to enter the transition state. But enzymes help speed things up quite a bit. Enzymes are proteins that are folded in a very particular way, so that they have a pocket called the active site on their surface. When enzymes get involved in a reaction, the substrate binds to the active site, and together they form the enzyme-substrate complex. And that helps stabilize the transition state. So enzymes decrease that extra energy required for the reaction, graphically turning our mountain into more of a hill. Think about this analogy. Imagine a little boy who's nervous about getting a vaccine, and he turns into a vaccinated child. That's the product. The transition state is where the needle goes in. And as you can imagine, the boy might get really anxious and upset, a highly energetic and uncomfortable state. In this scenario, enzymes are like adults who hold the boy and calm him down, reducing the anxiety or energy level of the transition state, and making the whole thing happen faster. Fortunately, enzymes don't get used up in the process. They attach to the substrate until it turns into the product, and then release the product. As soon as they're done, they find another substrate. What's more is that enzymes and substrates are like biochemical soulmates. Each enzyme is specifically designed for a particular type of substrate. For example, amylase is an enzyme in your saliva that specifically helps break down large carbohydrates into smaller sugar molecules that are further broken down by other enzymes. Now, the rate at which enzymes catalyze biochemical reactions is called enzyme kinetics, and there are two graphical ways to look at this. The first is the Michaelis-Menten graph, which has the concentration of the substrate on the x-axis and the speed or velocity of the reaction, which is how much protein is formed over time, on the y-axis. If there's a fixed amount of enzyme, the velocity of the reaction increases as more substrate is added. That is, until the active sites on all of the enzyme become saturated. At this point, adding more substrate won't do a thing, because there's no more enzyme to bind it. So the speed of the reaction plateaus. The point where the curve flattens out corresponds to the maximum velocity, or Vmax, on the y-axis. Now we can determine Km, which is the concentration of substrate at which the speed of the reaction is exactly half the maximum velocity. So we look at the y-axis, find what half of Vmax is, then we go parallel to the x-axis until we reach our reaction curve. From there, we go straight down toward the x-axis, and Km will be equal to that substrate concentration. The reason that Km is worth figuring out is that it inversely reflects enzyme affinity. If Km is low, only a little substrate is needed for the reaction to skyrocket up to half its maximum rate. So we're looking at an enzyme with high affinity. On the other hand, if Km is high, then it takes a lot of substrate to get the reaction to go at half the maximum rate. So the enzyme has low affinity for its substrate. Fundamentally, these graphs tell us about binding. In fact, the same graphic representation could be used to show how strongly drugs bind to receptors, or even transcription factors bind to DNA. So while we're here, let's look at how Vmax or Km can increase or decrease. Vmax depends on the number of enzymes that can catalyze the reaction. So increasing or decreasing the number of enzymes will increase and decrease Vmax. One way to increase Vmax is induction, which is when there's an increase in gene expression of an enzyme. The opposite, or a decrease in Vmax, might happen if there's repression or silencing of gene expression. If we're talking about membrane receptors or transporters, upregulation or downregulation of receptors or transporters on a cell's surface would cause an increase or decrease in Vmax, respectively. Finally, a drug might block enzymes, transporters, or receptors non-competitively and cause a decrease in Vmax. With non-competitive inhibition, the inhibitory molecule binds irreversibly to the enzyme active site, or reversibly to a different place called the allosteric site. The net effect is that substrates can no longer bind to the active site, and it's as if the number of available enzymes has decreased. So the effects of non-competitive inhibition are similar to what happens when there's less enzyme around, which decreases Vmax. Now let's look at Km, which varies according to enzyme affinity. Affinity depends on the shape of the enzyme and how well the substrate fits into the active site. So let's say that we alter the enzyme in a way that makes it bind more substrate. One way to do this is when a molecule called an activator binds to the enzyme and increases the enzyme's affinity for the substrate, lowering the Km. Conversely, competitive inhibition can be used to decrease the affinity of the enzyme. A competitive inhibitor binds to the active site instead of the substrate, and usually doesn't get metabolized, blocking the enzyme. Now, since this is a reversible process, adding more substrate means that it's possible to eventually outcompete and displace the inhibitor, but this increases the Km. Now, practically speaking, a common way to change the shape of an enzyme is through phosphorylation by kinases, or dephosphorylation by phosphatases. And many processes are regulated this way in our cells. In fact, the terms desensitization and sensitization refer to changes in a receptor's shape which decrease or increase its affinity for a drug. Now, let's switch gears and look at the Lineweaver-Burke plot. This graph is based on the Michaelis-Menten equation, which states that the initial speed of the reaction, or V0, is equal to the concentration of substrates times the maximum velocity, or Vmax, over the concentration of substrate plus Km. So here's where things get interesting. To get to our Lineweaver-Burke plot, we're writing this equation down as a double reciprocal. So flipping the whole thing upside down, we get 1 over V0 is equal to Km over Vmax plus 1 over Vmax plus 1 over the concentration of substrate. Now we can use this equation on the plot. 1 over V0 goes on the y-axis, and 1 over substrate goes on the x-axis. Conveniently enough, after flipping the equation upside down, Km over Vmax is represented as a straight line that intercepts both the y and x axes. The y-intercept is equal to 1 over Vmax, and x-intercept, since it happens negative to the origin of the axes, is negative 1 over Km. Therefore, the x and y-intercepts can be used to quickly identify Km and Vmax. The only trick is that, much like our reciprocal equation, now everything is upside down. So processes like induction and upregulation, which increase the Vmax, actually cause 1 over Vmax to decrease. So the line representing Km over Vmax would slope lower on the graph than the control. Conversely, processes like repression or downregulation or even non-competitive inhibition, which decrease the Vmax, will increase 1 over Vmax, which makes the line slope higher on the graph than the control. So interpreting a Lineweaver-Burke plot goes like this. Stimulation makes the straight line appear to go down. Inhibition makes it appear to shoot higher. Alright, as a quick recap. Enzymes lower the activation energy required for the substrate to enter the transition state. A substrate binds to the enzyme at the active site, forming an enzyme-substrate complex, and it's released after it's transformed into the product. There are 2 ways to look at enzyme kinetics, the Michaelis-Menten graph and the Lineweaver-Burke plot. Both can be used to determine the 2 kinetic variables, Vmax, or the maximum speed of the reaction, and Km, or the affinity of the enzyme for its substrate.
Farmacodynamics refers to the mechanisms and effects of medications within the body. Or more simply, it's what medications do to the body and how they do it. Alright, so in order to have an effect, many medications have to reach their target cells and bind to a receptor. Receptors are specialized proteins found inside the cell or on its membrane. When they bind to a signal molecule called a ligand, they can alter their shape or activity, which ultimately results in some change in the cell's activity or behavior. You can imagine the ligand as the key that fits into the lock, which is the receptor, causing it to open or activate. Now, depending on the effect a medication has on its receptor, they are often divided into 2 major categories: agonists and antagonists. An agonist is a medication that mimics the action of the signal ligand by binding to and activating a receptor. On the other hand, an antagonist is a medication that typically binds to a receptor without activating them, but instead decreases the receptor's ability to be activated by another agonist. Okay. Now, the maximal effect or response an agonist can produce, abbreviated as Emax, is determined both by the number of receptors bound to the agonist, which depends mainly on the amount of the agonist given, also known as dose, as well as its intrinsic activity, which is the ability of the agonist to fully or partially activate its receptors. Let's plot all this into a nice graph to show the relationship between the dose given, on the x-axis, usually on a logarithmic scale, and the response produced on the y-axis. So full agonists, upon binding to the receptor at high doses, are capable of producing a maximal response of 100% Emax on the y-axis. This represents the point when all available receptors are bound to an agonist. In contrast, partial agonists, even at very high doses, when they occupy all of the receptors, result in a smaller response, so their Emax will be lower. For example, a 70% response would shift the curve downwards. Now, let's say a partial agonist is used at the same time with a full agonist and they compete for the same receptors. If we increase the dose of the full agonist, it will displace the partial agonist from the receptor and the maximal response will still be achieved. So the dose-response curve will shift to the right without affecting Emax, but the dose required to achieve 50% of the maximum effect, also known as effective dose ED50, will be increased. In other words, when a partial antagonist and a full agonist of the same receptor are present together, then the full agonist's potency, which is the dose of agonist needed to elicit a maximal response, will be decreased, but the full agonist's efficacy, which is the maximal effect that an agonist can produce, will stay the same. Alright. At the other end of the spectrum, antagonists can be divided into competitive antagonists and non-competitive antagonists. Now, a competitive antagonist is a medication that reversibly binds to the same receptor site where an agonist binds, but it does not activate it. Competitive antagonists usually bind to the receptor in a reversible way, meaning that they bind and dissociate from it pretty fast. So when they unbind, it's more likely for one of the ligands to bind. So the inhibition caused can be overcome when there are more ligands floating around. This is sometimes referred to as surmountability. Now, on the graph, competitive antagonists typically shift the curve to the right without affecting Emax, but increase the effective dose ED50. So, competitive antagonists decrease the agonist potency, but do not affect the agonist efficacy. On the other hand, non-competitive antagonists don't bind to the same site as an agonist. They bind to a spot called an allosteric site. When they bind, it causes the shape of the receptor to alter in such a way that the ligand can no longer recognize it as a binding site. And even if it does manage to bind, it won't be able to produce the agonist effect. Now, binding of a non-competitive antagonist to the receptor is either irreversible or it dissociates very slowly. This means that the receptor will not activate no matter how many agonists are around. In other words, this type of antagonism is not surmountable. What we see on a graph is that the dose-response curve won't shift, so the ED50 won't change, but the slope and peak, and thus the maximal effect Emax that can be achieved by any dose of an agonist would be reduced. In other words, non-competitive antagonists reduce agonist efficacy without affecting agonist potency. Alright, as a quick recap, agonists activate receptors and can be divided into full agonists, which can produce a maximal response, and partial agonists, which can only produce a submaximal response. Antagonists prevent receptor activation and can be competitive, meaning they bind reversibly to the same agonist binding site, and non-competitive, meaning they bind irreversibly to the same agonist binding site or to a different allosteric site. Competitive antagonism can be overcome by adding more of the agonist, while non-competitive antagonism cannot.
Farmacodynamics refers to the mechanisms and effects of medications within the body, or more simply, it's what medications do to the body and how they do it. Alright, so in order to have an effect, most medications have to reach their target cells and bind to a receptor. Receptors are specialized proteins both on the cell membrane and inside the cell that can bind to a ligand. Now that ligand could be an agonist, which is a molecule that binds and activates a receptor. This means the receptor changes its shape or activity, and that gives rise to a signal cascade of intercellular molecules, the second messengers, which ultimately results in some change in the cell's function. Okay, now if we massively expose the receptors to their agonists, we'll get a huge downstream signal cascade and cellular response. But if we continuously or persistently flood the receptor with the same agonist at the same dose, what will happen is the ability of the agonist to produce that response will drop. This is actually a defense mechanism whereby cells prevent their overstimulation by agonists. If this happens very rapidly, like within a few minutes, it's called desensitization or tachyphylaxis. If this happens more gradually, like over the course of days to weeks, it's called tolerance. Desensitization can occur with the initial dose of a medication, while tolerance typically happens with repeated doses. Alright, so there are several mechanisms responsible for these phenomena. First, chronic exposure to agonists causes a decrease in the number of receptors. The decrease in the number of the receptors could result from the reduced synthesis of new receptors, also known as downregulation. Also, chronic exposure increases the degradation of pre-existing receptors through endocytosis, also known as sequestration or internalization. This is where the cell swallows up the receptor in vesicles, pinching off the cell membrane, and sends them to the lysosome for digestion. This phenomena can also be seen in chronic opioid and other drug users, where the target receptors are downregulated, so a higher dose is needed to achieve the same high. Alternatively, the receptors may be modified where they are uncoupled from their intercellular signaling pathway, meaning they can't activate the secondary messenger proteins anymore. This type of response is very rapid and mainly causes desensitization. When exposed to high doses of epinephrine, G-protein-coupled receptors, or GPCRs for short, get phosphorylated by intercellular enzymes called G-protein coupled receptor kinases, or GRKs. The phosphoryl groups then get bound by bulky proteins called arrestins, which prevents the receptor from activating their neighboring guanine nucleotide binding proteins, or G proteins. At the same time, arrestins tag the receptors for endocytosis, reducing the number of receptors on the cell membrane. In other cases, desensitization, or tolerance, arises from the exhaustion of intermediate messenger molecules. Take amphetamine, for example, which normally causes the release of amines like dopamine, serotonin, and noradrenaline from the nerve terminal into the synaptic cleft. Amine stores are limited, though, so continuous amphetamine use can lead to their depletion. As a result, amphetamine can no longer exert the same level of effect on nerve terminals with repeated use in quick succession. Another mechanism is the increased metabolic breakdown of the medication. An example is alcohol tolerance, where chronic use causes the body to increase production of enzymes that break down ethanol into inactive products. On the flip side, tolerance to nitrovasodilators, like nitroglycerin, is a consequence of their decreased metabolism, since their metabolite, nitric oxide, is the substance that causes the vasodilation. Lastly, the effects of a medication may be limited because of a physiological adaptation of the body. For example, diuretics like thiazides, which decrease reabsorption of water, could gradually become less effective in lowering blood pressure as the renin-angiotensin-aldosterone system increases its activity to maintain the blood pressure at high levels. Alright, as a quick recap, desensitization or tachyphylaxis refers to a rapid onset decrease in the effects of a medication after repeated exposure, whereas tolerance has a gradual onset. They arise from various mechanisms including a change in receptors, either downregulation or translocation, exhaustion of messengers, increased metabolic degradation of the medication, and physiological adaptation.
Farmacodynamics refers to the mechanisms and effects of medications within the body. Or more simply, it's what medications do to the body and how they do it. Alright, so in order to have an effect, most medications have to reach their target cells and bind to a receptor. Receptors are specialized proteins both on the cell membrane and inside the cell that can bind to a ligand and get triggered to alter their shape or activity. This gives rise to a signal cascade of intracellular molecules, which are the second messengers, which ultimately results in some change in the cell's function. Intracellular receptors are typically found in the cytoplasm or nucleus of the cell, and recognize small hydrophobic ligands, or water-hating ligands. These include molecules like steroids, which are happy to diffuse across the phospholipid membrane. Once bound to their ligand, the receptor-ligand complex attaches to specific DNA sequences that activate or inhibit specific genes. On the cell membrane are cell surface receptors, which are embedded into the plasma membrane and bind to ligands too large or hydrophilic to pass through. Hydrophilic meaning water-loving. Based on their structure and properties, cell surface receptors fall into three main types. First are ligand-gated ion channels, which form channels or pores that are generally closed, but then open up once they bind a specific ligand. They allow ions like chloride, calcium, sodium, and potassium to passively flow into the cell, down their gradient, and trigger the signaling pathway. Next are the G-protein coupled receptors, also known as 7-pass transmembrane receptors, which means they're really long proteins that have one end that sits outside the cell, and then the snake-like protein dips in and out of the cell membrane 7 times, and finally ends on the inside of the cell. The ligand binds to the end sitting outside the cell, and the end of the protein that's within the cell activates guanine nucleotide binding proteins, or G proteins, which contain an alpha, beta, and gamma subunit. Normally, the alpha subunit binds to guanosine diphosphate, or a GDP molecule, and the G protein is inactive. When a ligand binds to the receptor, the G protein changes shape, causing the alpha subunit to release the GDP and allowing a guanosine triphosphate or GTP to bind. This causes the alpha subunit to detach and trigger other proteins in the signaling pathway. Broadly speaking, there are three types of G proteins: GQ, GI, Broadly speaking, there are three types of G protein. This causes the alpha subunit to detach and trigger other proteins in the signaling pathway. Broadly speaking, there are three types of G proteins: GQ, GI, detach, and trigger other proteins in the signaling pathway. Broadly speaking, there are three types of G proteins: GQ, GI, and GS. Each type has its own kind of alpha subunit. The GQ protein activates the enzyme phospholipase C, which cleaves a phospholipid called phosphatidylinositol 4,5-bisphosphate into inositol triphosphate and diacylglycerol. On the other hand, protein GS and GI stimulate or inhibit, respectively, the enzyme adenylate cyclase, which takes adenosine triphosphate or ATP and removes two phosphate molecules and transforms it into cyclic adenosine monophosphate or CAMP. Inositol triphosphate, diacylglycerol, and CAMP then go on to stimulate and inhibit different sets of enzymes and molecular pathways. Finally, there are enzyme-coupled receptors. These are usually single-pass transmembrane proteins, meaning that they only have one transmembrane segment. The extracellular end of these receptors bind to ligands, and their intracellular end has enzyme activity. The enzymatic domain is usually a protein kinase, which phosphorylates other molecules. When a ligand binds, it triggers a conformational change in the enzymatic domain to form high-affinity binding sites for the second messengers. These second messengers get phosphorylated by the tyrosine kinases before heading off to activate other proteins in the signal pathway. Alright, so every cell has a unique combination of many different types of receptors. And in theory, each medication is specific to a particular receptor, just like each key fits into and opens a particular lock. This is described as a medication's specificity. In reality, though, no drug is 100% specific, meaning it could also bind to other receptors that are similar to its intended target, which is why all medications cause side effects. Now, the way a medication and receptor interact with each other is ruled by two main principles. Affinity is how strongly a medication binds to its receptor. This is mainly determined by the strength of the chemical bond between the two. The higher the affinity, the higher the potency, which is the amount of medication needed to elicit an effect. So, medications with high affinity can produce an effect at a lower dose. After the medication binds to its receptor, it must also be able to activate that receptor. This ability is known as intrinsic activity. Ultimately, the intensity of response to the medication depends on both the number of receptors bound to the medication as well as its intrinsic activity. The maximal effect the medication can produce is called the efficacy. Alright, so now let's plot all this into a nice graph to show the relationship between the amount of a medication given, also known as the dose, on the x-axis, usually on a logarithmic scale, with the response produced on the y-axis. What we get is an S-shaped curve, called the dose-response curve. At first, the curve is more or less flat, and that's because the dose is too low, so not enough receptors to bind to the medication to cause a significant response. As the dose increases, though, more receptor medication complexes form, so the response to the medication climbs upward. This is described as a graded dose-response relationship. Eventually, we reach a point where all the receptors get occupied, so the curve starts to flatten out. This is where the maximum response or effect, abbreviated as Emax, is achieved, which is a measure of the medication's efficacy. Now, if we move to the point where 50% of the maximum effect is produced, the effective dose of the medication producing this effect, known as ED50, marks its potency. So the smaller the ED50, the less it takes to get halfway of the maximum effect, so the more potent that medication is. To illustrate this, let's plot the dose-response curves of two medications, medication A and medication B, on the same graph. Notice that both reach the same maximum effect on the y-axis, so the same efficacy. However, on the x-axis, the dose ED50 required to produce 50% of that effect is much lower for medication A than medication B, meaning medication A is a lot more potent than B. In other words, the closer the curve is to the left, the higher the potency is. Now let's look at two other medications, C and D. Medication C achieves a higher maximum effect on the y-axis, meaning that it's more efficacious than D. In other words, the higher the curve, the higher the efficacy. At the same time, though, on the x-axis, the dose ED50 for 50% of that effect is the same for both, so they're equally potent. Alright, as a quick recap. Medications produce their effects by interacting with receptors, either intracellular or extracellular ones, including ligand-gated ion channels, G-protein-linked receptors, and enzyme-linked receptors. These interactions are characterized by the medication's specificity for the receptor, affinity or strong binding with the receptor, and intrinsic activity, or ability to activate it, which in turn determine its potency, or the dose needed to produce an effect, and efficacy, or the maximum effect the medication can produce.